Okay, question time, guys. So let me give you some information first about the exam tomorrow. There are 10 questions, and I have given half a page for each question. Okay, so it's not like 1 or 2 or 3 sentences. You may have to write something there. So one question is like, for example, what are oncolytic viruses? What are the different ways? I have made a question on oncolytic viruses. It was such a big hit. I was going to put a smiley face there, but then I did not. I said, well, the exam, you have to maintain some proper decorum for the exam. So one of the questions is on oncolytic virus. What are the different ways by which oncolytic viruses work? So of course, they attack the tumor cells specifically because they replicate in tumor cells, and then they form a bag and they lyse it, and then they activate the immune system. Then you can also add toxin to the wire and it will release it. So different way. So this kind of questions are there. So it's not like one sentence, what is oncolytic virus? There are 2-3 things in the same question. Every question is like that. Okay, in some cases I say, oh, give me an example of that. So I have tried not to become extremely specific about something, but if I ask you about something, I will ask you in more general terms. Okay, so that kind of questions are there. There are 10 questions. Of course, I could not put everything there. Like I cannot. 10 questions are not enough for this whole 30 lecture course. So, but I have, so few of your favorite topics may not be there, or few of your favorite topics may be there, I don't know, okay? So, it is like that. So, at least read all the important things. You should know the definition and understanding of all this. And in most of the cases I have said, give me an example to explain that, explain using an example. So I've tried not to say, okay, give me this example. So whatever it is, if it is oncolytic virus, take one example. If it is immune checkpoint therapy, take one example. If it is PGX, explain using one example. What is PGX? Explain using any one example. So at least have one example in your arsenal. If you're making someone understand, use one example. So that should help you. So pharmacogenomics, what is a nanobody? How does it work? What is a PROTAC? How does it work? This kind of thing. What is an antibody? How does ADC work? Versus simple antibody versus ADC, right? CAR-T therapy, tandem CAR-T therapy, kind of thing. I have specifically not asked you about when I was giving you crash course on immune system, because that's very complicated. So I have not asked you anything directly about explain immune system. But I have definitely included questions about the therapy targeted towards how will you target TME. So I have told you many ways of targeting TME, just any one is good enough. I just want to see that you guys got the gist of the course, okay? All of you are taking the exam tomorrow, right? You guys have attended all the time. Venkatesh has already said that he gets too many emails, I want to attend the course, but those students are not showing regularly, so he said they are not going to get the certificate. Okay, guys, ask me questions.
Ma'am, we have immunotherapy, then we have cell therapy. So in immunotherapy, we are using CAR-T cells engineered.
Please, वो कहाँ गया वो? In immunotherapy, so tell me what are the different things you can use in immunotherapy? All of you give me one immunotherapy thing. Yes.
Immune checkpoint.
Immune checkpoint therapy, very good, like PD-L1, PD-1, very good. And CTLA4, very good. आपका हो गया checkpoint, मतलब correct.
We can use oncolytic viruses or cancer vaccines.
Very good. That is also immune therapy, cancer vaccines and oncology, CAR T therapy, immune system. और कोई भी है क्या? क्या? Antibody drug conjugate. Very good.
That is what I'm T cell specific antibodies. Bispecific antibodies.
You can use bispecific antibodies. Very good. You can use bispecific antibodies. Yeah. You can use a PROTAC which will bind to a nanobody, which will bind to a tumor cell, and you can bring the drug there also. Very good. So go ahead. So what was your question?
I wanted to know that what is the difference between CAR T cell therapy or immunotherapy, specifically CAR T cell, and cellular therapy, because there also we are engineering the cells.
So CAR T cell therapy is a cell therapy because CAR T is part of a cell. But if you are giving when you say cell therapy, that means you are giving a cell. So CAR T or CAR NK or CAR M, they are all cell therapies, right? Because you are giving a cell. But if you are giving a gene kind of thing to turn on something, that is not cell therapy, that is gene therapy. And what was the other thing you were saying? Immunotherapy, if you are giving PD-L1 antibody, so that's not a cell therapy, that is you are giving just the antibody there. So it is a immunotherapy, but it is not necessarily a cell therapy.
So
but CAR T is a cell therapy, but it comes under immunotherapy. Yeah.
And then stem cell therapy can be included under cellular therapy?
Depends on that. Stem cell is a cell therapy, so it is a cell therapy. Stem cell is always a cell therapy. So you should know what are stem cells. I forgot all about stem cells, the pluripotent, the totipotent, the multipotent, the mesenchymal, the hematopoietic, and I definitely have a stem cell question. So what is a stem cell? How will you define a stem cell?
With potential to divide into different functions.
Self-renewal and differentiation.
So you have to also say the stem cells can make more stem cells. Don't forget that part, because you don't want stem cells to always only differentiate. Otherwise you will get, how will you keep the, So stem cells can make more stem cells, and then they can differentiate into many, many different kinds of cells. Yes. So that's it. So those are stem cells. And give me an example where you can use stem cell as a therapeutic agent.
In case of chronic inflammation, in order to avoid its like advancement into cancer, you will regenerate cell by giving stem cell therapy.
Tell me exactly how will you give stem cell therapy to prevent cancer from occurring?
In case of, I guess,
yeah, she's giving you that. Please take that. All of you remember to take that.
In case of IBDs or any like gastrointestinal inflammation, we can give mesenchymal cell to.
Very good. I had forgotten. Mesenchymal stem cells have immunomodulatory properties. Very good. I had forgotten. I was thinking myself, how can you stop inflammation? Yes, that's a very good one. Very good one. So see, you have chosen any. So you should have one example of therapeutic, whichever you like. Okay, one example of whatever. I had given you regeneration. You can generate the whole breast by that thing. Then bone you can regenerate, right? If somebody, you can have a cardiac patch, right? So things like that. So go through that, but just remember one part of everything. It's used as therapeutic, it's used as regenerative tool, it's used as...
Using the homing properties of the mesenchymal cells to include in the therapeutic part?
Yes, you can deliver the drug that way. Very good. So you can say, okay, I want to deliver the drug. I will use mesenchymal stem cells for homing property. You are done. Perfect. So you get to choose what you love the most there. That's how the questions are designed, as I recall. That's very good actually. Mesenchymals are really good ones. Then people who get leukemia, they get irradiation because their red blood cells are destroyed. So red blood cells are given from siblings with matching HLAs, right? So that is also use of stem cell therapy, right? You get new bone marrows. I don't know about this thing, but I've heard that taking out the bone marrow from a healthy person is very painful, and to give it. But but it is better than kidney donation. Less painful than kidney donation. What is a HLA?
Human receptor.
Leukocyte antigen.
Human leukocyte.
But what is it?
MHC sort of thing.
MSC equivalents. So HLA1, HLA class 1, HLA class 2. Great. I love our discussions so much. That's why I've included some things here and there as I learn about it. And liquid biopsy I was saying, oh wow, you can do liquid biopsy. That's really awesome. Because we don't want to open up the patient. So doctors are really against opening up someone because anesthesia has a serious side effect. I did not realize it. Anesthesia is so dangerous.
There is only one stage. If you cross, the patient will have paradoximal something. I don't remember it's stage of sleep cycle.
Okay, I was thinking doctors are overreacting. I never realized anesthesia is so dangerous. Like anytime they put you to sleep, they always make you sign some paper that you may never wake up from that. So that is very scary.
Also, ma'am, depending on the extent of how much you're opening up, the immunosuppression required is also massive, which is potentially risky in an already sick individual.
Exactly. But the good news is that it is shed in the bloodstream, and blood is least invasive. You know? I mean, taking out blood is a lot easier than anything else. It's just a prick of a needle that you have to suffer.
Still,
Right.
when we can do it with local anesthesia, it's okay.
Yeah.
When it comes to generalized,
That is very bad. That is very bad.
I have a weird idea that why can we use small molecule drugs in inhalation forms, like inhalers
Oh,
kind
you
of
do
form?
use it all the time. You
For
use
lung
all the
cancer, to
time.
be
Actually, it is written in one of the things there. So many drugs that you have to send it to the brain, they do not cross blood-brain barrier. But if you make it in an inhaler form, it will directly go to the brain. So that's a very easy way to get it to the brain, if you can make it in that way. So one of my friend was working in a company. Its name was something nasal. It was in Arizona, and it used to make many drugs which you could inhale, and it will directly go to the brain. So that's a very good idea, and it has been realized. See, keep in mind that you guys are very young and you're just learning about it. So for you, it's a brand new idea. But if it has been realized, then you should really pat yourself that you thought of the right thing, right? So your thinking is in the right direction, which is a big thing. Okay, so that's a very good point. And also, you guys should be very cautious of all the things that you put it on your skin, because it is directly going to your bloodstream. So we are very careful of what we put in our mouth, but you have to be equally careful of what you put in your skin and what are the things you're holding with your hands. Who was telling me about the fertilizers? Who was telling me? You were telling me about the fertilizers. See by hand how bad it is, right? Because it is directly going in the bloodstream. I talked to so many people during lunchtime and evening and this and that, so I get confused who was getting, because I'm getting too much input of information from all of you too, so I'm confused at the end of the day where it is coming from. You guys are living, at least as students, you are living in a nice place with a lot of greenery, so that is a blessing. if you get a job also in a clean, nice place where you are not exposed to dust. Go ahead, next question. We can even have 1 hour tutorial at the end of the day. We can have as long as you guys want. We'll just have as long as we want and we'll stop when we are done with it, okay? We can have 2 to 3, 3:30, okay? Next question. Today Vance is not there.
Ma'am,
Just take that thing, mic.
in the slide about CRISPR, like we were studying CRISPR-Cas9, there was a point which said editing the epigenome. So was that just using the targeting mechanism of the Cas to do the targeted demethylation and all that stuff, or was it something different?
If it is targeting the epigenome, you want to fix the enzyme which is doing that, because if you just remove the methyl group or something, that is
Okay.
post-translational modification.
yeah.
CRISPR cannot do that.
Yes, ma'am.
CRISPR can do it at the DNA stage only, right?
Hmm.
So it will actually target the epigenetic enzyme to have a long-lasting impact.
In CRISPR, if we are removing one particular gene in between the sequence, and then it will get like joined with any mechanism of DNA damage repair, then that new gene will be created at that site. Then will it? Then it may have its own like other expression.
So before you use CRISPR, the problem is already there, right? Before you use CRISPR. So you can remove the whole piece of there. So if it is BCR able, you can cut down the able BCR part, or you can remove the whole BCR able. If something happens afterwards, if you have removed the gene and some other part is going, it may not have any impact because the gene is missing. So what is that other part going to control there? But if it originates because you have done CRISPR, then you can go back and do the CRISPR again to remove it. So CRISPR can change the DNA sequence. CRISPR can remove the gene. CRISPR can add the gene. It can do a lot of things. So the other Gyan course that I taught actually about CRISPR also. But I kept CRISPR actually technology out of this because most of you already know how CRISPR works. So, and that is not directly related to this thing.
But we could specify with primers that what.
Exactly. It's all in the primers. The cgRNA that you create, you can exactly tell what exactly to do. It's a piece of cake to do it actually. Our collaborator, he created, now CRISPR vectors are available in AdGene, but that time they were not. So our collaborator created his student created a CRISPR vector, and we used it in our lab. And so they are all part of that paper, and it is just very easy to change any gene. It's very, very easy now. CRISPR is like everyday thing now. It's not a big thing. But it was a huge discovery. And we got the inspiration from bacteria. So bacteria are the one who have this CRISPR mechanism, and that is there to protect them from foreign attack. And that's what the scientists adopted. So Mother Nature has all the solutions. It's up to us to see the solution. Many times we see that if you are a Superman, you always find something which most of the people do not see. Superman does not mean like the superhuman doesn't mean just the strength. It means the smartness, right? So what this AI is doing, that when you show the pathology report, right, every pixel, it can read every pixel. It can see so many things in each pixel and do so many correlation compared to what we are seeing. So that's how it can see a lot more than what human brain and eyes can comprehend. Yes.
Retinoblastoma
Yes, it is.
So when its indeficiency causes
Yeah,
any
please
specific
give that. Just 1 second, repeat your question.
So, ma'am, retinoblastoma protein, you told us it is a tumor suppressor and its indeficiency causes some tumors. So, ma'am, are there any specific protein, are there any specific tumors which are caused by its indeficiency, like BRCA1, BRCA2 deficiency causes more breast cancers and ovarian cancer than other cancers? So, is there any specific cancer to
Yes,
this too?
absolutely. So retinoblastoma proteins are also known to be deleted in certain kinds of cancer. So when I was telling you about that, it actually specified which cancers it is specifically deleted. So when you do the cell cycle here, right? So when you're going through G1, there is a point where retinoblastoma protein is very important. So that's when it controls whether it should go past that point or not. So retinoblastoma protein is involved in cell cycle and it is deleted or mutated in certain cancers. But overall, a large percentage of cancer. Then there is another, P53 is deleted. There is another protein called P10 that is deleted. That is also a tumor suppressor. Then CDKN2A, remember? The MTAP one is a passenger which gets deleted by mistake because it is just next to it, like a bystander which gets deleted. So yeah, so tumor cells go out of the way to delete these proteins. So imagine how smart they are, like how smart they are. Somebody had Alzheimer's disease long time ago. So there was some kind of viral infection which led to Alzheimer's, and at that time it was not known that viral infection can lead to Alzheimer's disease long time ago. But the thing is that they grow, and they kill the patient. So ultimately, they are live cells, but they all die. Same thing with cancer. It is taking so many different pathways to survive, but you kill the host, the cancer also dies with the host. But the thing is that it is what it is. Prevention is the best medicine. That's the message here. So let me ask you, if you have to summarize the course, I will ask you guys, what are the things that you learned? And it is personal to all of you, right? So you tell me, what are the things you remember that you learned in this course which made sense to you? Like, I attend a course and I have some take-home message. I attend a lot of courses which are different from these courses, right? And then somebody said, okay, what did you learn in this course? And if I have to say it in 1 or 2 sentences, it's different from everyone else. So what are the things that you learned that you didn't know before? I'm going to go around and ask everyone. CAR T cell therapy. So those are the things you really learned about it. What about you?
Almost everything.
Almost everything was new to you. Yeah. Do you remember something like obviously you cannot remember the details, especially for these kind of crash courses, but what are the things which resonated most? Stem cells, very good. What about you?
Ma'am, before joining this course, I had a little bit of idea of everything, but I was not having the clarity that which one to prefer, why to prefer, where to prefer. So now it's a clear picture
of all the things. And then the thing is that you have the notes. You can always go back and read the notes. Okay, so that's the most important thing. That is your thing. It is all in one place. What about you?
Pharmacogenomic was a new concept, and product,
These are very new for you. Actually, before I prepared this course, I did not know about pharmacogenomics myself because that has not crossed my research thing. And I was saying, oh, now it makes sense why I did not respond to that drug. So it is very strange. I have a chronic headache problem, And one time I was given a medicine for chronic headache, and that medicine was okay, but I didn't realize that I was very quiet when I was taking that medicine, and somebody close to me said, you know, you are always very depressed, not depressed, matlab down, not energetic. And that medicine is not supposed to be, it's antidepressant side effect, but I was depressed. And I did not know that, and somebody commented that, and then one day I actually forgot to take the medicine, and I was just so chirpy that day. All day I had a lot of energy. Then I never took it again. So its side effect was antidepressant, and I was taking for headache. My doctor was horrible. She was prescribing all these stupid medicines to me. But I got depressed with that. So my pharmacogenomics is different, and I did not know about pharmacogenomics till I prepared for this class. So that was my learning too, pharmacogenomics. Yes, go ahead.
Ma'am, basically I have a major in immunology. So I mean, yeah, it's core of a discipline in which I am interested in. So
Oh, please give that. It has, yeah. You have a major in immunology.
yeah, so
Coming from that immunology background, all of the things which I can relate to made it easy for me to learn more of the concepts. Also, the questions I've asked is something which it's basically my imagination coming to the reality and getting the real time answers and resolving it. It's a new thing for me, and it's really amazing.
Thank you, thank you, thank you. Your turn. Oh, yeah, you can go ahead. You have the power. Just 5 minutes में आ रहे हैं, हाँ 5 minutes में आ रहे हैं.
So I am also interested in immunology, and I've always looked at disease proliferation and been very interested in the mechanisms from the side of the disease. But in this course, drug delivery and like the methods of approach to the disease have been so fascinating to me that I think I'm more interested in that part now.
That is very
Because
good.
I used to think from the side of the disease now I'm thinking from the side of the human being.
Exactly, that's very good. Go ahead.
Ma'am, pharmacogenomics was a new concept and precision medicine.
Precision medicine, of course, we did not know actually what is the meaning. I did not know actually what's the real difference between personalized and precision. Precision seems more precise, so I thought, you know, you go from personalized to precision. I learned, no, you go from precision to personalized. That was a wake-up call for me too. Yes, go ahead, Vanshika.
Before I only knew that conventional therapies of cancer, but I got to know immunotherapy, which was very interesting for me, and it was very fun process learning all this. And as he said, it was very fascinating to know so much new things that we can approach towards cancer.
Yeah.
Before I used to think that it is a uncurable disease, like we cannot cure it.
It is
so
not.
I got to know many new things.
Yeah, you got to, and you guys are going to contribute to many new things, so it will become completely curable. Go ahead.
My answer is kind of similar to hers. Before the course, I only knew that cancer can be treated, or not even treated is not right word, but the response to cancer can only be chemotherapy or surgery. But now I know how many different aspects to it. I got to know there are different types of tumors and they should be treated differently.
That is also
Epigenetics was a great what should
introduction
great
to that.
introduction to that. It was very
Yeah.
fascinating thing.
Yeah.
That same genetics but different expressions.
Different expressions. That's
Yeah.
very good. That's very good. Yes, yes, yes.
And about the cancer and immune cells interaction, that how
Oh, that
smartly
is-
cancer controls immune cells. That was very
a very
fascinating.
new thing too, immunosuppressive environment.
Immunosuppressive.
Yeah, that is very good. Thank you. Go ahead.
Ma'am, for except the AI part and drug repurposing and nanoparticle part, everything was new for me because I am currently working in both all the three topics, and I have also worked in the nanoparticles. I have assisted someone making transdermal patches. So for me, the cancer part was like new. Everything was new for
So
starting
you
from
are
the
in
therapy.
which lab?
Ma'am, in LP University.
Which university?
LP University, lovely
LP
profession.
University, you have worked on nanoparticles.
Yeah,
and
there I have
everything.
assisted someone.
That is very good.
Yes,
That
ma'am.
is very good. But now you have a more complete
Yes.
picture there. So the bottom line, the last message is there is that it is just constantly evolving. And I've not even covered all the topics. I've covered, whatever I thought were important, but it is much beyond that. But at least you have enough background that you know what questions to ask ChatGPT, right? Because you have to also know how to use it, right? Because it is just so that is very good. What is the difference between polypharmacy and polypharmacology?
In polypharmacy, we have multiple. In polypharmacy, we have multiple drugs for one target. You can say combination therapy.
It is a combination therapy for different targets,
Okay.
not one target.
Okay, and but in case of polypharmacology, we use one single drug which hits different target.
Very good. That's the difference between polypharmacy and polypharmacology. Acha. What about Gleevec? Is it a drug made for different targets, or is it one drug against many targets?
Initially, it was made for one specific target, but accidentally it worked.
It was an accident pharmacology for Gleevec. That's when they realized polypharmacology is very good. Acha, what is deep medicine? Precision medicine plus AI, not that. Very good. Acha, how will you define CAR-T therapy? Anyone?
Enabling T cells with CAR receptors so that they can target the tumor cells, recognize tumor cells while not attacking the normal body cells and not get attacked by the body's immune cells.
That is very good, but would you like to add something more to it?
It's sort of a platform which brings antibody directly in close vicinities with the target.
CAR-T,
It has nothing to do with antibody. CAR.
genetically engineered T cells.
These are genetically engineered T cells which have SCFP receptors, so nothing to do with the antibody. Just be cautious when you are writing the answer. Don't be that generic because it would be wrong. So you have SCFB receptor that binds. See, just be very accurate, Venkatesh. So it will recognize tumor cells by binding some tumor antigen. Okay, so make sure you say tumor antigen, and then once it binds, then it has the co-stimulatory molecules and other things, so it will spray. It is not going to bring antibody there. It is it is weaponized, so it will spray gran enzyme, perforin, and all those things. So it will spray and kill it. Okay, so it doesn't depend on something else coming and killing it. It will just spray it. So make sure be cautious in answering that. So look at it. Okay, so CAR-T, polypharmacology. So tell me 3 or 4 different ways of how will you do active targeting. Tell me different ways of doing active targeting to tumor cells.
Folate.
Huh?
Exploiting folate receptors, drug with folate.
Very good. So that's one. Second,
Active transport, so nanobody.
again some specific target, but nanobody coupled to something, right? Coupled to a drug. Very good. And that so make sure you write nanobody raised against a specific tumor antigen, tumor specific antigen. So write it properly. Okay, you may know it, but I have to know that you know it completely, right? So that is okay. What is a PROTAC molecule?
It is used in degradation of
Degradation of undruggable.
It links between gene of interest and to the E3 ubiquitin.
Protein of interest.
Protein of interest.
Protein of interest, not gene of interest. Gene is DNA. Protein of interest on one end. So write it properly. So it binds to the ligand. It has a small molecule which binds to the protein of interest. Write it properly. And that protein of interest, the linker is there, and it is not E3 ubiquitin. That would be wrong. E3 ubiquitin ligase. That is a ligase. E3 ubiquitin is no word. E3 ubiquitin ligase. Okay, so that is there, and it will bring it. So how is it superior or how is it advantageous to use a PROTAC as compared to small molecule? And they degrade it. So you are also taking care of protein-protein interaction with that one, right? So, okay, that's very good. और क्या-क्या चीज़ हम लोगों ने पढ़ी है? Give me the topics and I'll ask you.
G protein.
G proteins, right? So how do you define a G protein? Define a G protein.
It is involved in secondary pathway. Secondary
Intracellular signaling protein.
GTP dependent secondary messenger, secondary.
Define G proteins in 2 sentences. आप लोग जो है ना गलत कर रहे हैं. हम अगर बहुत लिबरली भी पॉइंट्स देंगे तो भी नहीं दे पाएंगे इस केस में. तो आपको एक्यूरेटली बोलना है. देखो, वी आर साइंटिस्ट, वी हैव टू आंसर वेरी एक्यूरेटली. सो व्हाट आर जी प्रोटीन्स? हाउ डू दे गेट द नेम जी प्रोटीन्स? आपको ट्राई मारना है. G proteins are those proteins which bind either GTP or GDP, and when they are bound to GDP they are turned off, and when they are bound to GTP they are turned on. So G proteins are, this is the definition. G proteins are molecular switches which bind to either GTP or GDP. When they are bound to GTP they are turned on, when they are bound to GDP they are turned off. That's the definition. So G proteins means they have to be bound to either GTP or GDP. So don't say second messengers or anything. That comes a lot later. That is not a definition of G protein. G proteins are GTP or GDP binding proteins, and you have to specify GTP on, GDP off. Okay, and you also should say it's a molecular switch. Okay, so that's very important. That was a good question. What do kinases do? Phosphorylate by using? What is the difference between kinase and a phosphorylase?
Phosphorylase add in inorganic phosphate.
Very good. They are not using ATP. Good. That's the difference between phosphorylase and phosphate. And how does CTLA4 works? What is happening in that immunotherapy? That is the hard one. PD1, PDL1 is easy. Explain to me PD1, PDL1 first. Anyone?
PD1, PDL1 are the normal immune checkpoint where PD1 binds with PD1 of the T cells binds with PDL1 of antigen presenting cells, and it turns it off like if it is hyperactive. But some tumor cells as defense mechanism express PDL1 and interacts with T cells PD1, turning them off.
Very good. It is perfect answer. 100% correct. बहुत अच्छा. हाँ. So you know CTL4A, how does it work? That is a complicated one. If you can remember.
Yes, there are 3 signals.
There are 3 signals. Achha, I can give you a hint here. I have not asked you about CTLA4 because I thought वो थोड़ा hard हो जाएगा, बहुत कुछ याद रखना होगा, पर आप मुझे बताइए, at least आज बता दीजिए.
CTLA4 is expressed in T cell when they are hyperactive. The tracks have B7 receptors which binds with PTLA4.
CTLA4.
CTLA4 turning down the activity of T cells.
So this is very good. So what happens is that under normal circumstances, T cells, after they get activated, then they will express CTLA4, and then APCs are the one which will have the B7 which will bind to CTLA4, right, and turn it off. But in tumor cells, they will recruit Treg cells which will bind there and turn the signal off. So this is totally correct. So what you said is totally correct. Okay, so CTLA4. So these are immune checkpoints. So what are immune checkpoints? You can explain using PDL1, PD1, right? So what is synthetic lethality? Explain using one example, any one example of your choice.
BRCA1 or in BRCA1, BRCA2 mutations, PARP inhibitors are used.
Yeah.
Like synthetic lethality, one gene is not working and we can block the other gene, so the overall cell dies, like tumor cell.
So why is it called synthetic lethality then? You have to explain, don't take the shortcut. Explain. Go ahead.
Because in cancer progression, one gene is already defected and we are targeting another gene, we are causing the lethality.
By that, but normal cells have both genes there, so if you inhibit it, it's not going to affect. So explain everything. Okay, तो थोड़ा सा ये बता दीजिएगा कि normal cells में both pathways A and B are there, so if you inhibit A, B takes over, if you inhibit B, A takes over. But in tumor cells, one pathway is already defective, so if you hit the second one, cells die. That is synthetic lethality. Correct? और कौन कौन से टॉपिक्स हम लोगों ने कवर किया था?
Therapies and hallmarks.
हाँ, थेरेपीज एंड हॉलमार्क्स. हॉलमार्क्स तो पूरे समय बोलते ही रहे हम लोग, है ना? I can tell you I don't have any question on microbiome. तो थोड़ा सा रिलैक्स कर सकते हैं आप लोग.
And on sporadic diseases, do you have?
क्या?
On sporadic and
Yeah, so I want to ask you, genetic and sporadic diseases, what does it mean actually?
Genetic diseases are inherited genetically from the
Germ cells.
from the germ cells, and the sporadic diseases are the ones that we acquire during the.
Exactly, that's what it is. Okay, that is what it is. And most of the diseases are sporadic. And what is exposome?
Exposome could be any external factor which influence the kidney.
Exactly. And so sporadic diseases are caused by exposome. Okay. आप लोग के लिए ना लाइफ लॉन्ग लेसन है ये कि टेक केयर ऑफ योर एनवायरनमेंट. नोबडी इन द वर्ल्ड, इवन द रिचेस्ट पर्सन जो कि बहुत ही बिजी होता है, नोबडी इज बिजी इनफ नॉट टू टेक केयर ऑफ योर सेल्फ. यू कैन just always have to take care of yourself, because you know life is it's very strange. It seems like अभी तो शुरू हुई है और फिर बहुत जल्दी ख़त्म भी हो जाती है ना? तो मतलब देखिए, जितने एज तक रहना है जिसको, one has to be completely healthy, right? You cannot be a walking मतलब unhealthy person. That's what I'm saying. So the take home message from any such course is that really take care of yourself. It's not a joke. Next thing, I like that you do the exercise thing, body. That is very good to hear. I'm really happy to hear that thing. People are much more jagruk these days compared to in our time. People were never doing exercises there. at that time. People didn't know, but you know, at that time there was no computer, so people were always walking around doing the job. So exercises हो ही जाता था सबका. There were not so many cars. It was culture that you walk to close by places. And it was not considered social symbol or something because it was the habit, you know. So we used to get the right amount of exercises anyway. सब लोग ग्रुप बना के स्कूल जा रहे हैं तो it used to be fun walking, even going to school, you know. So and things were safe and everything, है ना. और फिर we used to play, मतलब इतना physical activity because we were not playing on computer, we were just actually playing. और इतना अधिक खेलते थे ना कि वो सारी एक्सरसाइज बिल्कुल हो जाती थी. And we were just climbing up and down stories, ऊपर नीचे ऊपर नीचे ऊपर नीचे ऊपर नीचे पूरा दिन दौड़ते. So and that is why now exercise is important because you know in IIT Kanpur we didn't have electric vehicles, we had to walk or bicycle से जाना होता था, मतलब साइकिल से, है ना. जोर से हाँ, biomarkers, it's very important to remember all the biomarkers. You do have a question on biomarkers, so you should remember all sorts of biomarkers. That's a very important one, I forgot.
Drug resistance, therapy resistance.
Yeah, read about therapy resistance also, biomarkers also. I did not know that you guys are going to get the grades also, because I was thinking it is pass and fail, so I was relaxed कि सब लोग तो पास हो ही जाएंगे. But I would not like to give you a bad grade, so please do well, okay? I would feel very bad किसी को B देना पड़ा तो, है ना? I'm going to be as generous I can, but I still have something that if you write something wrong, I cannot make it right. I have to respect that thing. I'm too old-fashioned for that. Okay, so please do very well. Ask me as many questions you want to ask me today.
Do you have something with cancer vaccine?
जोर से.
No. I'm asking about question. Do we have something in question cancer vaccine also related to?
Could be. I don't remember, honestly, I don't remember. Usually what I do is that I will tell you one or two things which is not there, like microbiome, but I don't even remember. I'm not so fond of microbiome. I mean, it is extremely important, but not an important topic. I don't know what to ask you about microbiome, that's the thing. Epigenetic gene therapy is very important. Know about all the different mechanisms. Tell me epigenetic mechanism. I know, Venkatesh, you are very good in epigenetics. I have noticed that. And you get it very quickly. You propose a lot of things epigenetically. So tell me.
If two persons have similar genetic makeup for the same substrate, but they have different expressions of protein, it might be because of their epigenetics. Epigenetics is the expression of the same genomic material because of the condensation pattern of the genetic material of the DNA.
Yeah,
the
so
tightening and loosening of the DNA
due to which epigenetic modifications? So make sure you say methylation of DNA, which is acetylation. Methylation closes it. So here is the thing between you and me, right? So we may have the same epigenetic mechanism, but within us also, depending on our environmental conditions, epigenetics can change. So for example, because of stress, epigenetic pattern may change. So it is not that it is different between different people. It has nothing to do with it. It is all within us that at different times we respond to different things. So I attended a neuroscience meeting maybe 10-15 years ago. So there the new finding was, which is now a very old finding, that how do you make short-term memory versus long-term memory? Can anyone tell me? Some memories are long-term, right? हमेशा याद रहती हैं, and some memories are short-term. So people who have Alzheimer's, they retain long-term memory, but they don't retain short term memory. They will forget अभी 2 मिनट पहले क्या हुआ था या कल क्या हुआ था, but they will remember their kids. So what happens in the brain? You probably don't know, it is epigenetic modification. Those memories which are stored, somehow the proteins which store are methylated, or they are differently epigenetically modified, and those memories become long term because of the epigenetic modification, and short terms are not. By the way, robotics in AI, deep medicine can also help, like you think of something and the robotic arm will move. So जिनके prosthetic limbs होते हैं, तो उसमें खाली सोचने से वो limbs को आप move कर सकते हैं. जैसे हम लोग सोचते हैं और सब कुछ कर लेते हैं, है ना, तो उसमें खाली thought से you can move it. So that is where AI is going.
So do they integrate PNS with prosthetic arm?
I don't know how they do it, but it is thought controlled.
PNS is peripheral.
I think somehow they do it. I don't know. I have no idea. I was reading about it this morning actually when I was talking to my niece. So, because she's very much into neuroscience. So, and how thoughts are arranged in the brain in the 3 dimension, and how do we comprehend 3-dimensional space.